Regulating gene expression. Table of contents. Mutations. Regulating prokaryotic gene expression. Regulating eukaryotic gene expression. Mutations. A mutation is a permanent change in the DNA sequence of a gene. Mutations in a gene's DNA sequence can alter the amino acid sequence of the protein encoded by the gene. Somatic mutations occur in somatic body cells and are passed on by mitosis, but not to sexually produced offspring. Germline mutations occur in germline cells that give rise to gametes, meiosis. A gamete with a mutation passes it on to the new organism at fertilization. Mutations may or may not affect the phenotype. Mutations are the primary source of genetic variation. Mitosis and meiosis will be covered in more detail in a following unit. Point mutations. Point mutations occur when one or more nucleotide bases are changed, added, or deleted. There are 3 types of point mutations: substitution, insertion, deletion. Point mutations may result in proteins that are less efficient, but maintain enough function that the phenotype is not changed. Or amino acid substitutions may not affect protein function. For example, substitution of one hydrophilic amino acid for another. Substitution mutations. When a nucleotide in a gene is copied incorrectly during DNA replication, one nucleotide can be substituted with another. This results in the incorrect amino acid sequence, changing the structure of the protein. So in green, you see the correct DNA sequence at this point as AAA, which means the RNA transcript would be UUU, and the polypeptide that UUU codes for is PHE or phenylalanine. What happens if we have a substitution error at this second position where instead of AAA, we have ATA? Our RNA transcript will be off by 1 base. We would have UAU, and that codes for the polypeptide tyrosine. We can think about the DNA sequence of a gene as a sentence made up entirely of 3-letter words. So we have this sequence here that if I split it up into the sentence of individual 3-letter words, I would read, "The sun was hot." So what is a reading frame shift? If this sentence represents a gene, then each letter corresponds to a nucleotide base and each word represents a codon. If you shifted the 3-letter reading frame, it would result in a sentence which is not understandable. So look what happens if I shift it. I have blank blank T, HES, UNW, or if I shift only one, then I get partial word here and a whole bunch of stuff that doesn't make sense if I shift the reading frame. When a nucleotide in a gene is copied incorrectly during DNA replication, a nucleotide can be added or deleted. This is an insertion or deletion mutation. And when that happens, a reading frame shift is what results, and we get the wrong amino acid sequence, which changes the structure of the protein. So let's look at 2 different examples. In the first, we have our correct DNA sequence, which you can see codes for 1, 2, 3, 4 amino acids. If I insert another A here at the beginning of this sequence, the first amino acid, phenylalanine, UUU, is the same. But because I put an A here, everything shifts down 1. I get this reading frame shift so that instead of AAA, I get the U that shifts, and I get UAA, which is a stop codon. And so the polypeptide would stop here. We'd miss all of these last 3 amino acids. Now, what if we insert What if we have a deletion mutation? That was an insert mutation. What if we delete one of the A's from this first 3 of the DNA sequence? If I delete it, then I get a transcript that's UUA instead of UUU, which is leucine. And then I have AAG. Now take a look at this. You see how this is AAG? This is AAA. Those 2 codons code for the same amino acid. And so does GGG and GGC, glycine here. So this first amino acid is incorrect. The next 2 are correct, but due to the reading frame shift, because I shifted everything to the left one when I deleted, I don't get an amino acid for this last 1, 2, and then I'm missing a base here for this last codon. And so again, I have changed the structure of the resulting polypeptide. Silent mutations. A mutation is silent if it has no effect on the protein coded by the gene. The redundancy in the genetic code makes this possible. Each amino acid has more than one possible codon, so if a substitution occurs, the same amino acid may still be coded. This reduces the possibility of a mutation located in a gene causing a change to the protein. So take a look at these 3 amino acids and look at the different codons that code for that same amino acid. So that if I just substitute one amino acid if I have a substitution of just one base, it's quite possible that it codes for the same amino acid. And that would be what's known as a silent mutation. Let's look at an example of a silent mutation. Valine can be coded for by 4 different codons. Only the last nucleotide is different in all 4. The codons for valine are GGU, GGC, GGA, and GGG. If the original DNA strand has GGU in a particular location coding for valine, as you see here, and there's a point mutation right at this 3rd base where A is substituted for the U, there will be no net effect on the amino acid coded for. The sequence remains the same despite the mutation. Therefore, the mutation is silent. Mutagens. A mutagen is a physical or chemical agent that can change the DNA of an organism and thus increases the frequency of mutations. Mutation in germ cell lines is the raw material for natural selection and evolution. Somatic mutations cannot be inherited, yet, if they accumulate in one cell, they can lead to diseases such as cancer. Only germ cell line mutations can be inherited and potentially impact future generations. Mutagens include ionizing radiation like X-rays and gamma rays, ultraviolet waves like sunlight, alkaloid plants like tobacco, coca, and poppy, sodium azide, a component in many car airbags, and benzene, a solvent used in plastics and synthetic rubber. Not all mutations are caused by mutagens. Spontaneous mutations occur due to errors involving changes in the chemistry of the DNA or DNA replication, repair, and recombination. So in this diagram, we have a strand of DNA, C's and G's, that during the replication process, one of the strands here slips out of place. And during the DNA repair process, we come in and replace a missing C and a missing G. Okay? And in doing that, there's 9 bases here, here, and here. One is just out of place. But in the repair process now, we have an extra C and an extra G, which will result in a mutation. Are mutations always bad? Not necessarily. Very rarely, a mutation will cause an individual to become stronger than the rest of its population. Sometimes a mutation can give a prokaryote resistance to a new antibiotic or toxin. These would be advantages to the individual, and they may become better able to survive in their environment. Mutations are a major contributor to genetic diversity, which fuels evolution. Harmful point mutation example. The human gene TP53 encodes the tumor suppressor protein P53, which normally inhibits the cell cycle. A gain-of-function mutation, in other words, a mutation that makes it more effective in TP53, causes the protein to promote the cell cycle and prevent cell death, which increases the proliferation of cancer cells, a gain of oncogenic function. The sickle cell disease allele differs from the normal one by one base pair, resulting in a polypeptide with only one different amino acid. The shape of red blood cells is changed, preventing them from moving smoothly through the bloodstream, causing oxygen shortages for the body cells. It causes pain, swelling, frequent infections, vision problems, and other critical symptoms.